All right, we're going to look at, just briefly, I don't expect you to memorize this. This is pretty much what I just explained to you, the sexual life cycle of animals. But we're also going to look at plants and fungus. And what I really want you to focus on is, this isn't the greatest picture in the world. They show meiosis here. So each individual person's cells go through meiosis, either in the testes or the ovaries. And that makes eggs or sperm, which we call gametes. And that happens no matter if any of these people ever have any children. But occasionally, what's very rare is the fusion of the egg and the sperm. So here they're showing the egg is right here. They're showing it with 3 chromosomes, just to simplify it. The sperm has 3 chromosomes. And then you have the fertilization. So fertilization is the fusion of the 2 cells. That makes the zygote, which you can see has the 6 chromosomes all in one cell. So, so that's the sexual life cycle, that's the new individual. And then that zygote grows up into either a male or a female and, becomes part of the process again. But meiosis by itself is not sexual reproduction. Meiosis is the first step, the cell division that makes the gametes. The fusion, the fertilization, is the second step, which makes the zygote, which is the next individual. That's really important because sometimes students think that meiosis is the whole thing, but no, it just makes eggs or sperm. That doesn't, that's certainly not the whole thing. One thing I want you to see in this picture, let me erase all my ink here. In this picture, the way they've done it is proportionate. The amount of the picture that they devote to the main part of the body being diploid. Most of the life cycle in animals, most of the body is made of diploid cells, and there's really a small group, just the gametes, that are haploid cells. All right. Now look in the next picture. This is plants. You will look a lot more at plant sexual life cycle if you take Biology 1407, which is a really good class. And in this picture, it's showing you've got meiosis. Some of the cells go through meiosis. Eventually, there's fertilization, so those are there. The details are a little different in between, but you have this alternating meiosis and then fertilization. But this is showing you, just in a very rough sense, that there's actually, it's about 50-50 in plants. There's a whole plant that is made of diploid cells, and then there's a whole nother plant. This sounds weird, but this is in plants, this is how it is. There's something else made out of haploid cells. And so the life cycle is different, but in terms of how the bodies of the organisms are structured. But the meiosis makes the haploid cells. Fertilization is the fusion of those haploid cells. And then in the fungus, look at this. Look how much of the fungus body is diploid. The picture isn't showing it, but I'm saying that this slice here kind of represents how much of the life cycle, how much of the body is spent, or is diploid cells. And most of the life cycle, most of the organism is actually made of haploid cells in fungus. So when you see, for example, a mushroom, which is a kind of fungus, or mold, they use bread mold. When you get that moldy piece of bread at the bottom of the bag, that most of what you're seeing there is the structure, it is the body of the organism, but it's mostly made of haploid cells. So, but you still have an alternation of meiosis, and then here they wrote nuclear fusion, which is the concept. The fusion is the fertilization. All right. So the same concepts are alternating there. It's just there's some different details in between. All right. You do not need to memorize the life cycle, all the terminology in the life cycle of the fungus. You do not need to memorize the terminology in the life cycle of the plant. You do need to know the animal because that's the one I went over earlier. But I just want to show you that all of these organisms have meiosis and fertilization, but some of the other parts of the details of those groups are different. So you don't want to assume that everything's the same. The way it is in the animal kingdom is exactly the same in the plant kingdom or in the fungus. But meiosis and fertilization alternate in the sexual life cycle of all three of those groups. All right, so we're going to go into the features of meiosis, the details of meiosis. So the big picture is that meiosis is the cell division that makes the gametes, the haploid cells. And that's the first step of the sexual life cycle or the sexual process, sexual reproduction. But now we're going to delve into the details of meiosis specifically. And I think when you take anatomy, they go into some of the details of fertilization, although that is simpler to explain, I think, the fusion of two things. It's an interesting topic, actually, but we're only going to cover the details of meiosis in detail. All right, so you have, actually, meiosis has two nuclear divisions. And you'll remember mitosis, it really only had one nuclear division per cycle. So meiosis is not a cycle, it's just a straight line to a dead end, I guess. Not a dead end, but to an end, the gametes. Once the gametes form, once a cell goes through both divisions, the product cells are done. They can't divide, they can't go through again. They can't go through meiosis immediately again. And so they're basically waiting for a fertilization event. All right, so the two divisions, the first one's called meiosis 1, the second one's called meiosis 2. Meiosis 1 occurs first, meiosis 2 occurs second. And it seems silly that I would say that, but you guys know that sometimes things aren't as obvious as they should be. So meiosis 1 is first, then meiosis 2. And each of these divisions each of these meiotic processes is subdivided into subphases. So meiosis 1 has prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2 also has prophase 2, metaphase 2, anaphase 2, telophase 2. So those are the same, virtually the same names that we used for the subphases of mitosis. But in this case, you have a number after the phase. So if it's prophase 1, if there's a number there, it has to be meiosis. If I just say prophase with no number, I guess that would be vague, but if there's a number, it's definitely telling you that it's meiosis. So if it's prophase 1, it's part of meiosis 1. If they say prophase 2, it's part of meiosis 2. And so that number helps you to know that we're talking about meiosis. And sometimes it's a Roman numeral, sometimes I see it just as a Arabic number. So it could be just a number 1 or a number, numeric, a 2, or it could be Roman numerals. So that doesn't matter. All right, in prophase 1, which is really the first part of meiosis 1, the chromosomes, homologous chromosomes, remember I said in a diploid nucleus there's a copy of chromosome 1 and another copy of chromosome 1. And what they will do in meiosis, which never happens in mitosis, by the way, is those two copies of chromosome 1 will find each other, which is kind of amazing. Like how? The nucleus is a crowded place. There's all kinds of other chromosomes in there. The chromosomes do condense, just like they would in mitotic prophase. So the chromosomes condense, they're already replicated. The replication happened right before meiosis began. And so the chromosomes are in their dyad form, and there's two copies, two dyads. and each dyad has 2 sister chromatids. So 2 dyads of chromosome number 1, and they will come together, and that's called synapsis. And then there's 2 copies of chromosome 2, and they will come together, and 2 copies of chromosome 3. I mean, can you imagine the chaos in the nucleus where each copy of a chromosome is trying to find the other one just like it in this crowded space of 46 chromosomes? I mean, the fact that it works, and it has a pretty low failure rate, is kind of amazing. And so the coming together of all these different chromosomes is called synapsis. It happens in prophase 1. This is an event that never happens in mitosis. So you'll remember that we talked about focusing on the differences between meiosis and mitosis. This is a big difference. In prophase 1, the homologous chromosomes find each other and they stick together very closely. And because they're in the replicated form, when you have 2 dyads that come together, when they're touching, we call the whole grouping a tetrad. And I have a picture on the next slide to show you. And so a tetrad would be, so this is a dyad version of a chromosome, the red one, and the blue one is a dyad version of the homologous chromosome of the same type. So this could represent 2 copies of chromosome 1 coming together, or it could represent 2 copies of chromosome 15 coming together, or 2 copies of chromosome 22 coming together. Now, what's interesting is if you have a cell that has an X and a Y, so a male cell, a cell from a male, if you have a male cell, the X chromosome and the Y chromosome are actually not the same length, but they will still come together, which is kind of amazing too, like how, So we're not going to get into the details of how that works. There's a lot of research that has gone into figuring out how do the chromosomes recognize each other. It's a chemical recognition. But they do, and they stick together like this. And the picture on the left is more realistic of how they probably lie together. The picture on the right is usually the way it's actually shown in a textbook, because it's more two-dimensional, where they put the homologous pair next to each other, but really they probably lay on top of each other like you see on the left. But on the right, you can see the crossing over, the exchange. I usually call it the exchange of equal corresponding segments. All right? It's like if two humans of the same height, this is not going to be a perfect analogy, but if 2 humans of the same height cut off one leg at the knee and swapped it, and then somebody sewed it back on, this would be a weird Frankenstein experiment, but they're exchanging equal corresponding segments. At the end, they still have, 2 knees, 2 feet, but it's not exactly one of their legs isn't exactly the one they got out of bed in the morning with. Now they got somebody else's leg, but equal corresponding segments. And the difference is that what genes are on that part of that chromosome, it's the same gene. So like here they show gene A, and when I say it, you can hear it. Gene A, gene B, gene C. But the blue chromosome might have a very slightly different version of gene C than the red chromosome of the same type. They both have gene C at the tip of their chromosome, of that chromosome, but they might be a slightly different version, or they could be the same version. And so a crossover just switches them, and so still each chromosome has one copy, or has A and B and C. It's just that the versions change a little bit. This creates a little bit of genetic variation at this moment, and then when these chromosomes are passed into the gametes will be a little bit different. All right, so that synapsis is the coming together very closely, and then the crossing over is the exchange of equal corresponding segments. Typically, you have an exchange between 2 of the chromatids that are not sisters. So like a blue one and a red one are not sisters. If you had a crossover that involved sisters, it wouldn't really matter, because remember, those are identical. So you could have a crossover. We think that sisters can cross over, but it doesn't create anything interesting. So we kind of ignore it. But when there's a crossover between non-sister chromatids, then you get a little bit of a different combination on those chromosomes now. All right, so that's the first step. So meiosis 1 starts with prophase 1. The other things that happen in prophase 1 are the things you would associate with prophase in general, meaning the nuclear envelope disappears, the chromosomes condense, but it's this synapsis and this crossing over that's unique to meiosis 1, to prophase 1. Now, that is the very first step. Now, what meiosis 1 ultimately is going to achieve, by the end of meiosis 1 by itself, what we're going to achieve, we will start with one cell, one diploid cell with replicated chromosomes at the beginning of meiosis 1. By the end of meiosis 1, we will have 2 product cells, and each of those cells will be haploid. So the parental cell, and by parental I just mean the starting cell, the cell starting meiosis. It doesn't have anything to do with like that person's parents. It's their cell, but their cell that starts through meiosis inside their body, inside their reproductive organs, their cell goes through meiosis 1. You will initially have 2 product cells or 2 daughter cells that are haploid. At the end of meiosis 1, the 2 product cells are haploid. But the difference is that the cells are haploid, the daughter cells are haploid, but the daughter cells will still have chromosomes in the dyad form. And so the second division will simply take cells that have chromosomes in the dyad form, and it will pull apart the dyads and create daughter cells that have chromosomes in the monad form. The second meiotic division is much more like mitosis. The second meiotic division is more similar to mitosis because you just start with a cell that has dyad chromosomes. That's not working. Similar to mitosis. You start off with a cell that has, or 2 cells that have dyad chromosomes, and by the end of meiosis 2, you'll have 4 cells, and they'll have monad chromosomes. But the ploidy of the cell will stay the same. All right? So let me outline this here. Let me see if I can get I wish I had I could do typing. Yeah, okay. All right, so meiosis 1 starts with a cell that is diploid. Oops. Previous. Why can't I get my pen? Okay. Meiosis 1, you have one cell, I'll put one circle, that's diploid, 2 sets of chromosomes. So if this was human, it would have 46 chromosomes. And the chromosomes, each chromosome is in the dyad form. So those are 2 different issues. 2 sets of chromosomes in the cell. Each of the chromosomes is in the dyad form. Oh, this is really bad. Why is my mouse doing this? Try different. I think it's just the drawing is always bad. Okay, let's say diploid. I'm going to say DI for diploid, DY for dyad. No, it's not any better. By the end of meiosis 1, that's one cell. By the end of meiosis 1, you have 2 cells. Each of the cells is haploid. You'll have to write this in your notes. Haploid. But each of the cells still has dyad chromosomes. And then those each will divide. And by the end of meiosis 2, you will have 4 cells, and each cell will be haploid, and each cell will have monad chromosomes. So if this is human, you start off with, in the very beginning, you start off with a diploid cell, so it has 46 chromosomes in the dyad form. By the end of meiosis 1, you have 2 cells, each has 23 chromosomes per cell, and so that's haploid. But the chromosomes are still in the dyad form. And in by the end of meiosis 2, you have 4 daughter cells, 4 product cells. Each of them has 23 chromosomes, and the chromosomes are in the monad form. So sorry about the scribbly drawing, but I think I got that point. All right. So what happens in prophase 1? Like I said, the same things that you would think about in any prophase. The chromosomes condense, the nuclear envelope disappears, the spindle forms. That's standard even for mitosis. But the synapsis and crossing over is the key difference here, so put a star next to that. In metaphase 1, the tetrads, remember the tetrad is 2 dyads that are synapsed. So you have 2 dyads that are synapsed for chromosome 1, you have 2 dyads that are synapsed for chromosome 2, you have 2 dyads that are synapsed for chromosome 3, etc. All of those groupings move to the metaphase plate, move to that equator line together. And the way the spindle attaches at this time is different than mitosis. In meiosis 1, one fiber attaches to the dyad chromosome on the left, and one spindle fiber attaches to the dyad chromosome on the right. And that happens for each of the tetrad groupings. And so in anaphase, when those spindle fibers pull apart, it moves a whole dyad to one side of the cell or the other. Let me show you here. Okay, so here, I skipped ahead a little bit, but so here is the way, let's say you have the blue and the red chromosome together. Let's say those are both copies of chromosome 15. And that grouping there, that synapsis, is at the metaphase plate. Can you see how the spindle has attached to the blue chromosome from the left side of the cell, and the spindle has attached to the red chromosome from the right side of the cell, so that when it pulls apart, it pulls those dyad chromosomes away from each other. So that's the way anaphase 1 will separate the chromosomes. Anaphase 2 will be much more like mitosis. One fiber will attach to the sister chromatid on the left side, one fiber will attach to the sister chromatid on the right side, and when those fibers pull apart, you actually have the sister chromatids ripped apart. So the dyad chromosomes become monads. So that's really the critical thing that's happening in the first meiosis compared to the second. Now, there is a consideration, we call it independent assortment. It's the fact that whenever you have the chromosomes in metaphase 1, and both of these pictures show metaphase 1, they show a tetrad of large chromosomes here and then a tetrad of smaller chromosomes here. Just for simplicity, they don't try to draw too many chromosomes. But this tetrad on this top picture, you have the large blue chromosome on the left side of the screen, more towards the left, and then the red chromosome a little more on the right of the cell, I guess you could say. And the same for the small pair. You have the small blue chromosome on the left and the small red one on the right. But how is that different? Look at this picture here. It's also possible, because remember, you don't just have one cell that goes through meiosis, you have lots of cells. They all start off the same, but then once they go through meiosis, there's some different possibilities of things that could happen. One possibility is what I put a star next to. You could have all the blue chromosomes lined up on the left and all the red chromosomes lined up on the right. Now, in reality, they're not colorized, but they're just doing that so we can differentiate between them. And remember, the blue chromosome might have a big C allele and the red chromosome might have a little C allele. But on the bottom picture here, what you can see if you look closely at this lineup, the blue on the big chromosomes, the blue chromosomes on the left and the reds on the right, just like before. But look at this bottom pair. Do you see how the red one is now on the left and the blue is on the right? And there's no reason why it couldn't be that way. Sometimes the cells line up the way the top picture shows. Not the cells, sorry. Sometimes the chromosomes line up within the cell, something like what the top picture shows. Sometimes they line up the way they are in the bottom picture. So, it doesn't matter. As long as they get to that midline, it doesn't matter. But what's going to happen is if they line up with the blues on the left and the reds on the right, then by the end of both divisions, you'll have the gametes will have blue chromosomes in some cells, red chromosomes in other cells. But if they line up the way the second picture shows, then you can see here the different possible outcomes of the gametes. This gamete has a large blue chromosome with a small red, and this one has a large red with a small blue. So those are all fine. It's all perfectly legit. It's just that depending on how those chromosomes line up left to right, will determine for that particular cell what the gamete outcomes will be. And you'll remember that some of the chromosomes have a big C allele, some have a little c. Maybe on the smaller chromosomes, some of them have a big Q allele and some of them have a little q. And so you'll get these different, slightly different combinations of traits, ultimately, the genes for the traits in the different gametes. So when independent assortment happens, it's just a random process. It's kind of interesting. There's 8 million possible kinds of gametes that any one human individual can produce. Slightly different gametes in terms of like what kind of traits they carry, just because of this, just because of independent assortment. I know here they only show 4 types, but we have 23 pairs of chromosomes. So independent assortment gives you all these different possible alignments of chromosomes and gives you all these different, slightly different possible gametes. That's why you and your sibling don't look exactly the same, because the gametes, the eggs weren't exactly the same and the sperm weren't exactly the same, even though you have the same parents. All right. Telophase 1 is fundamentally, there's nothing real special about telophase 1. The nuclear envelope reforms. We have now just one copy of each type of chromosome in each cell. The cells do go through a cytokinesis, although it's not usually mentioned, which splits into 2 actual daughter cells. From telophase 1, at that point you have 2 daughter cells, or 2 maybe product cells, but they're not done, because those cells will immediately go into the next division, which is meiosis 2. Now meiosis 2 is much more like a mitotic division. At this point, each cell, if this is a human situation, each cell would have 23 chromosomes. The starting cell in meiosis 2 would have 1 set of chromosomes, so one copy of chromosome 1, 1 copy of chromosome 2, 1 copy of chromosome 3, etc. But the chromosomes are still in the dyad form. And so at this division is where the dyad chromosomes are going to be pulled apart, and you'll end up with monad chromosomes in the daughter cells. So if we start meiosis 2 with 2 cells with dyad chromosomes, we're going to end meiosis 2 with 4 cells that have monad chromosomes. And we already went over this. The way the chromosomes in anaphase 1, the dyads, 2 dyads are pulled away from each other. In anaphase 2, the 2 sister chromatids that make up any one dyad are pulled away from each other. All right. So we want to focus a lot on what's different about meiosis. All right. You've studied mitosis. You had a test on it, but how is it different than meiosis? In meiosis, you have synapsis and crossing over. It's in prophase 1. That doesn't happen in mitosis. All right. Number 2, anaphase 1 involves the separation of homologous chromosomes from each other, not the separation of sister chromatids. And in mitosis, you never have the separation of homologous chromosomes. That's not a thing. And then number 3, there is no DNA replication that occurs, after meiosis 1, but before meiosis 2. However, for mitosis, there is a DNA replication for every time, every cell cycle. All right, and this is a side-by-side. Hopefully in your book you get a clearer picture. It doesn't come out that clearly, but mitosis, if you start with a diploid cell, you end with 2 diploid cells. Or if you start with a tetraploid cell, you end up with 2 tetraploid cells. Whatever, in mitosis, whatever you start with, you end up with 2 of those at the end. So mitosis is just about making more of genetically identical cells of the same type. But meiosis has nothing to do with that. Meiosis, if you start with a diploid cell, you end up with 4 haploid cells. And those haploid cells cannot go again through meiosis. So meiosis is not a cycle. It's just it's part of sexual reproduction, but in of itself, it's not a cycle. Because these haploid cells, these are the gametes, and like I said, most of them are just going to die. Now, if any one of these, and it's not that there's just 4, but for any one cell starting meiosis, you get 4 product cells total. But most of these will die, and then occasionally you'll have one that gets fertilized with another haploid cell from another individual. So that's the fusion. All right. So maybe spend some time looking at this figure, but use your textbook because this is really blurry. So my final statement is that meiosis makes haploid cells. In animals, these are the gametes. Like I said, in plants and fungus, it's a little different. The immediate product of meiosis are called spores. They are haploid, but then they eventually form into the gametes. But we're not going to focus so much on those in this course. But in animals, the haploid cells will, we say, mature into the gametes. And then they wait for fertilization. I would recommend that little animation that I put on that's in your sapling is really very helpful.